All right, now we're going to look at antibodies, right? So we've learned where the antibodies were from, what are their functions, right? So we already learned all this main functions, right? So what kind of cells secrete antibodies? B cells, right? And what type of B cells? B cells aren't the specific type, right? It's a specific subcategory. And so it breaks down, differentiates into these plasma cells, and plasma cells are the ones that actually generate these antibodies, okay? Let's look at the structure now. So if I were to draw an antibody structure here, so in yellow, this is the heavy chain, all right? And then you have in pink, this is the light chain. Okay, so if I were to write it like for pink, this is light, and then yellow is the heavy chain. So we're just generally looking at the actual structures of what's present and what's not present, all right? So in yellow, we said it is the heavy chain region of the antibody, all right? And if I grab some blue, these are held together by these structures for the tertiary structure of the protein called these sulfide bonds. So there's these disulfide bonds that hold the light chain and heavy chain together. And then when we look at the upper region versus the bottom region, let's use a different color here. The region that doesn't change, we call that the constant region. And the region that changes, which is the more upper region, this is the variable region. And in the variable region, what's really important is this is where majority, if not all, the antigens bind to this antibody, right? So this antibody comes in here. Let's use blue again. This antibody is present here, and you have this antigen coming here, binding to that specific receptor. That specific receptor is binding. And that actual region of where the antigen binds to the antibody, for example, if I make a little circle here and shade it in, this is called the epitope. So the epitope is the region of binding of the antigen to the antibody, okay? Epitope, right? And so what happens is whenever the antigen binds down, there is a reconfiguration of the actual structure, what makes it selective for that antibody in order to generate some kind of response, whether activation of a certain cell type, certain mechanisms of agglutination, where the cell bundles up and becomes sticky. You have neutralization, where the opsonization, where you have the actual inhibition of the receptors, and also precipitation that takes place, right? So this binding here causes the actual antibody to reconfigure, and that region is specifically the variable region, right? And so that's just the basic structure, right? And all antibodies have this function, right? When we talk about the different types of antibodies, right, there's three main structural differences. You have pentamers, dimers, and monomers, right? And so the different names is indicator of the number of binding sites. So a pentamer has 5, a dimer has 2, and then a monomer has 1 binding site, right? So not all antibodies look specifically like this, right? But their receptors is something that they all share, okay? So if we look at the different types of antibodies that we have, let's see, what's the best color? So we go first, let's go IgA. So Ig, what does that stand for? Immunoglobulin, right? So antibody, another name for it is immunoglobulin. IgA's main responsibility is located in the majority of the mucosal layers, right? These are all in the mucosal layers of the intestines, the gut, any kind of saliva, right? Tears. And the way to remember this too is in breast milk. And so the way I always remember this A is the accept. So the baby accepts IgA from the mom, right? So if you have problems remembering this, you can always remember IgA is responsible for accepting, okay? Next one, IgD. Okay, so IgD, remember dirty or diseased, right? But disease kind of makes it a little confusing, right? Because you're like, oh, these are some kind of disease, right? So let's just say dirty, so some kind of bacteria coming in, some kind of microbial. IgD stimulates your basophils and your mast cells to secrete histamine and also a lot of antimicrobial proteins, right? They're also present in certain receptors in your B cells, right? What else do we have here? And what's really important is IgA is a dimer. IgD, E, and G, the next we're going to talk about, are monomers, and IgM are pentamers. Right? So IgE, so the best way of remembering E is for like eosinophils, right? So these stimulate not only the basophils and the mast cells, but also the eosinophils, right? So they generate during allergies, any kind of allergic responses, any kind of asthmas, right? And also parasitic worms, right? So parasitic worm was selective for these eosinophils. And then we have IgG. This is also another monomer. So monomer. So the green is monomers. The pink is a dimer. And then the next color I'll use, whichever color I decide to use, I don't know yet, is going to be a pentamer. Okay? So IgG, think give. I always think give. This is the only antibody is able to cross the placenta, right? So across the placenta and it goes into the baby. So IgG is also a lot of good indicators of a lot of infections, right? So it's very passive. This is the example when we talked about passive immunity. That specific antibody that goes through the placenta is the IgG, which helps prevent infections that are generated from any pathogens for the baby. Okay, and then finally the IgM in yellow, and we said this is a pentamer. Pentamer means what? Means 5. So you have pentamer, you have 5, and this is unique about it. When I always remember this one, I always remember it's many. Many for many binding sites, right? And it's really important to understand this term I'm about to present here. It's called avidity. So you've learned about affinity before. Affinity is the amount of attraction given on one receptor. So the binding of one ligand to a receptor, the actual potential binding ability, right? That's affinity, right? One receptor, the frequency of binding, the potential of binding. The next term I want you to understand is avidity. Avidity is the number of receptors that are able to bind, right? So the more receptors you have for the potential of binding, the more avidity you have, right? So for example, if I give you an example here, and use up all the colors. What other color? Blue. Here. If I were to ask you a question like, true or false? True or false? The IgM has a greater avidity than IgA. This is a true or false statement. This is a true statement, right? Because remember, IgM has 5 binding sites, a pentamer, and IgA has 2 because it's a dimer. Okay? Or another question, for example, would be like, does IgE have higher avidity than IgA? No, right? Because IgE has only 1 receptor, whereas the IgA has 2. So IgA has a higher avidity than the IgE, than the monomer. So dimers, so if we were to actually categorize them here in terms of avidity, here, I can put it here. You have the pentamers in terms of avidity, increasing avidity, that go to the dimers, which then eventually goes to the monomers. Okay, and so that's basically showing you the actual comparison of avidity.